you. Thank you everyone for coming. And so I'm going to talk about identifying Compton jets in loop extrusion and replication processes. So this is a bit of an overview. I'm going to do a background and then the proposed algorithm. I haven't even talked about the algorithm itself, but basically finding jets and then some preliminary results. So for the background, really, I think I have to start with 3C based assay. So this is the Hi-C Rao et al. 2014 protocol, and essentially you're able to identify regions of the DNA, pairwise interactions, and genome-wide. So there have been some chromatin elements. I'm being quite vague, but these chromatin elements have functional implications. So really, we have some kind of open and closed regions. We have a bit more specifically promoter-enhancer interactions. We also have these kind of transcriptional hotspots that have been viewed in different kind of perspectives. So to be a bit more specific about these chromatin elements, we have loops. So on the left is what you would have in vivo. So and on the right is the contact map, so the data that is generated by Hi-C. And so you have kind of two parts to it. So you have the cohesin ring kind of holding the DNA together, and then you have the CTCF. So this is a protein that blocks the ring from extruding. It binds to a specific sequence to the DNA so that it kind of stabilizes its loop in time. That's a loop. And then this is a TAD. So you might not always have this the cohesin CTCF bound here, but it's really like a region where you just have kind of random ligations, random interactions, and then you also have a stripe. So this is kind of defined temporally. So you might start by interacting here because there is no CTCF that blocks on one side. So you might start interacting here and just kind of sliding along as the DNA kind of protrudes through the ring. And so this is the pattern that you see in Hi-C. And then topic of this talk would be jets. So you can kind of see what's going on. You have kind of these rings loaded on, no CTCF, and it generates these kind of lateral interactions that kind of come up as a secondary diagonal. So finding these quantum elements, so you can't really read this, but there's a lot of loop callers and there's a lot of TAD callers. So that's the first and second, and then the third is stripe. So there's a little less, but for jet callers there's even less. So there's about 4. Actually, the fourth one isn't exactly a jet caller, but it can be used for a jet caller. So then, what are these jets? So they're also called fountains, plumes. So they're a relatively new structure again, found in 3D assays. And it's again characterized by it looks like a hairpin, basically, the DNA. And so there's kind of 2 contexts in which jets have arisen. Okay, so I'll go over the first one, just a cohesin model. So this is the recap of the loop that we saw, but kind of in like a nice animation. So we have the cohesin model where jets have been identified in cohesin. So this is quite interesting. Cohesin DP, double positive mouth thymocytes in 2022, and they were initially kind of hand identified, so 37 genome wide, and they implicated the loading sites, which is the tip, to be small active regions on the genome. So this cohesin model has been found by other kind of studies. So this is a depletion study. So they essentially knocked out 2 proteins, CTCF and WAFL. So WAFL unloads the rings. So if you knock out what is unloading the ring, you should have more rings loaded onto the DNA. And then if you get rid of the CTCF, which blocks the ring, then you should kind of get that multiple rings loaded on. So we see that jet pattern occurring with the auxin. So I just want to get like an appreciation of the diversity of jets. There's a lot of very recent papers that have kind of shown these jets occurring in kind of strange cell lines. But yes, so we see these jet-like patterns. Then there's also the replication model. So replication process creates jets in bacterial cells, and this is quite well known and characterized. You know, in bacteria, you have kind of the replication fork 2, and this generates these kind of lateral interactions that end up as the jet-like pattern in Hi-C. But then the question is, what about eukaryotes? Is it the same? And so in 2024, there was a new variant of the 3C Hi-C called Repli-HiC, basically enriches for interactions associated with replication. So, and they found about 2,000 jets genome-wide in human K562 and mouse embryonic stem cells. So that's a bit of the background. Now I'm going to talk about, you know, the current methods and challenges in finding these jets. So there's two main ways to view Hi-C data as an image and a graph. People have done both, but I'll be focusing on an image. That's kind of what the current methods and previous methods have done in terms of jet finding. So the existing algorithms, I'm just going to kind of group it together. These three, they essentially work by a kernel convolution method. So you have a sliding window, so it's not exactly convolution, but basically convolution. You're going to have a sliding window down the main diagonal because that's where these jets are found, near the main diagonal. And so you have this one kind of zoomed in region, and then you would take an inner product, so some kind of like similarity function. There's others that are used below, but you must specify the kernel. So you must have it look like a jet in order to kind of have that high similarity, right? So going to give a number in one region. And so if you do a sweep, then you should get a vector. And then you can do the threshold. So some limitations on these methods is that they can't really capture the diversity of jets, specifically the size, thickness, the asymmetry, the curvature, as well as kind of maybe some combinations and things that are just even difficult to put in words. I don't really know how to describe the differences, but they do look different. And this will correspond to having to like a completely new kernel each time you want to find these jets. So what kernels do they use in the previous methods? They use one from polymer simulation, Isiaka, and Fontanka requires you, in fact, that you hand identify a couple of these jets. And then they form the aggregate, the mean kind of kernel. And then that's how you get the kernel. But there's also limited training examples. So there's only about 37 at the time of development. There was 37 jets hand identified in just one cell line, so it's quite difficult. We did some preliminary benchmarking on the 37 jets, so Fontanka and Isiaka, and they show variable overlap between the methods. Maybe Fontanka does a little bit better with 14 of the true set that is identified. And as of runtimes, we see that Isiaka takes a very long time. That's because the polymer simulation takes quite time. But we see that Fontanka is relatively quick. So this is the third algorithm, and I don't have to really go into the detail here, but you can see that we have some assumptions about the jet. It protrudes diagonally, 45. Maybe you have some leniency in the length and the width, perhaps, but in cases of curvature, we can clearly see that there are some kind of constraints on the imposed by the algorithm. So we would like to develop an algorithm that doesn't really impose these strong assumptions on jets. As multiscale, not machine learning, we had limited training samples, and that was kind of fast. And so in order to answer the first question, we had to answer what is the essence of a jet? Something that kind of holds it together. So this is where I'm going to go into the proposed algorithm. So the idea is you view the image as a surface on the XY plane. So this is the Hi-C visualized as a surface. It looks pretty cool. And this is a jet. Okay? And you can kind of get a grasp through these pictures how a jet looks like. And why does that help? Well, I'm going to make a claim here that jets are ridges. And ridges are actually a feature that's quite prominent in old conventional image processing. So you have ridges that kind of vary in size. They can go in any direction. And it's quite a flexible feature. And it leverages concepts from differential geometry. So what exactly does that mean? So this is a very simple ridge. It's a very simple ridge. We have a surface that's defined by I, so that's the image. And let's say I have a point O on that surface, and I kind of want to quantify this now. I want to quantify it. So I want to have a number that is high at this point because we're on a ridge at this moment. So we have the top view on the left and right. And so we zoom in. I'm going to make a tangent plane, and then I'm going to define like an arbitrary vector in that plane. It's just completely arbitrary. And then I'm going to kind of slice that surface in the direction of the V. And I would like to get the closed form solution for this blue line. This is the top view of the blue line. For the X and Y components, it's pretty straightforward. You just multiply it by a T, so that's where we're parameterizing it. And then the different T, you can describe the complete blue line, X and Y at least, but not the Z. But the Z is relatively straightforward. We have the surface function I, which takes X and Y. So I just plug in the X and Y, and I should get the height. So voila, I have the blue line as a function in R3. This is, I'm going to call it F. So I'm going to kind of look at that F and the T, and imagine that I do a proper shifting. So I can just shift, add a constant, and I might get something like this. The reason why I do this, remember, we're trying to quantify this. So what if I take derivatives? So the first derivative and the second derivative. And the second derivative, specifically the magnitude of the third component, so that's the Z, will give me a kind of a good measure of ridginess. So we do the math. So F, you take the derivative, second derivative, and you look at the third component, take the absolute value, and that becomes my number, essentially. It's a bit kind of technical, but bear with me, it gets a bit more simple. So we have, again, that kind of expression. If I express it as matrix vector, it's in a bit of a familiar form. Some of you might recognize it as the Hessian matrix. And then now it's even more recognizable now that this is the quadratic form. So kind of going back, my choice of V was actually arbitrary. So I'm going to say, what if I chose V to be parallel with the x-axis? So looking like this, top view would look like this. Then intuitively, my parabola would be much more thinner, and that would cause the second derivative to be a larger value. So we would want that, right? We would want this number to be high because this is a ridge. We want this number to be high. So this is an optimization problem. We want to maximize that across all the V's in my tangent plane. That kind of maximizes that ridgeness quantity, which is expressed as a quadratic form. And this is a very well-known optimization problem that's given by the largest eigenvalue of the Hessian. And with that, you can also compute some other interesting things you might have seen in class as well. So with the theory, we can now form the basic building blocks of a ridge detection algorithm. So we have a convolution block. This is convolution with the kernel that I have as an image. So you have the inputs, some Gaussian blurring, and then you essentially compute the directional derivatives. So these are standard image processing filters. You essentially compute the derivative kernel, and then you convolve it, and yes, this is the output that you should get. And for each kind of X, Y position, I extract the values, and it's three values, three numbers, and they will form my Hessian matrix. It's because it's symmetric, only three. So you can see it's a 2 by 2 matrix. And I can just do eigen decomposition to get my lambda one, which is the richness number, and the eigenvector, which is I show as a vector field. It's a little small, but yes. And you do this for all the X and Y's, then you get an output map, which is what we see here. So from the building blocks, we can now form a simple ridge detection algorithm. So we start with the previous output, do some non-maximum suppression, then some thresholding, and then you connect the dots. So this is a very simple standard image processing pipeline. The actual algorithm has a lot more bells and whistles to this. You need a lot more tricks to actually get this to work and to work well under noise. So we will be relying on an ImageJ2 plugin that is an implementation of Carl Steger's algorithm. And this is the ridge detection algorithm we'll be using when I mention from now on. And so these are some results on the Hi-C data, and you can see that it detects jets that have a certain kind of curvature to them. So we kind of addressed this first point, but now I'm going to address the multiscale part. I haven't really talked about that. So now the idea is, high-level ideas, we have ridge detection. We want to kind of combine it with scale space theory. This has been done before by Tony Lindenberg in 1998. So this is a motivating example. So this is a blob detector, a Laplacian, but you don't really need to understand that. We're just going to look at what it does. So this is a butterfly, and as you can see, there's various blobs, sizes, and if you do a blob convolution over this, we should detect these blobs. So typical procedure being that we have a Gaussian blurring, and then we do the blob detector. But the Gaussian blurring actually has this, the standard deviation, right? So that's the shown here. So what if we vary this? And that's the essence of scale space. You vary this, and you see what you get. And so this is kind of a movie showing different sigma values. And for the lower sigma values, the smaller blobs are emphasized. The larger sigma values, the larger blobs are emphasized. And you can kind of see this is the essence of the scale space, how it works. So yes. There has been papers published that utilize this framework. So loop calling has in fact used this mustache to find multi-scale loops. So loops are defined by one position, and so the extension to ridges is not really straightforward. So we're kind of building up now the algorithm. So imagine a jet that has diffuseness. So it starts kind of small, but then it becomes wider and wider. And this is a specific example where scale space will be useful. What we do is we do Gaussian blurring at various scales. Then we do the ridge detection, the image J that I mentioned. And this is actually a very key part of the scale space. You need to normalize your filters. I'm not going to go into too much theoretical detail here, but essentially you have to multiply the derivative filters by a certain kind of, by a number. And we get, the output for the sigmas 1, we get a lot of noise actually in our output kind of ridge map. But at sigma 1.4, we see that maybe the beginning part is emphasized. And a little bit more at 2.7. By the time we get to the end, we get the diffuse part to be captured as well. So we see scale space kind of working as a building block. And so proposed algorithm is that you take the output, the eigenvalue maps, this is across scale. This is a tensor, and you impose some conditions, now some binary conditions. So if you recall, this is that function f. We just want it to, be a local maxima. So we have these simple conditions here that ensures that it is a ridge. And so that would give me these kind of x kind of points in my tensor. actually visualized. This is what it looks like. So you can see that the scale increases. These are the points across scale as the z-axis. And ridge detection will also give me the actual coordinates. So these are the XY coordinates. These are the positions. And so along the blue line, along the positions, I can track what is the scale that has been assigned to you. And so it's continuously increasing. And so from this is a bit arbitrary, but we take the median. We take the median scale, and it seems to work well in practice. So in this example, it's about 8. And so we just now focus exactly on this, just one output map, one standard deviation blurring. So we take that one output map, we compute saliency, and we get a ridge rank, and that's it. So how do I compute saliency? So I'm going to represent the line, my ridge line, as X, Y, and Z, but Z is the scale. So it's a set of points. Now I'm going to compute this. So it's it looks complicated, but it's just really a sum. Okay, it's just a sum. I take the across the values, I just sum the values of this matrix that I kind of index X, Y, and Z. So we modified this function a little bit to impose some constraints specific to the jet. So we would like it to be in the direction 90 degrees, because that is the direction of the jet, especially if we rotate the Hi-C. And we can get the direction by the eigenvector. So this is the theory for those of you who are interested. I'm going to skip this for the interest of time. This is the theory of the scale space ridge detection. So as a summary, and we've built everything up. So we have the input image, we have the scale space Gaussian blur, then we have the ridge detection, and we have the saliency, which we did for the angle to be between 80 and 100 degrees. And then we have finally a set of ranks. Yes.
So what is the like one sentence situation about the saliency thing?
Sorry, what's that?
Or what is the one sentence situation of the saliency?
Saliency is a sum.
The sum.
It's just a sum.
Okay.
Yeah.
So we are summing here for what? Different point.
So this gamma is a ridge. It's a set of points defined in X and Y and Z. And it takes input that one ridge and just sums across the values of the tensor.
So is it comparing between different ridges?
No, this is independent.
Okay.
Yeah. So some preliminary results. We tested it on 2 cell lines. And this is the top ranking in Repli-HiC K562. And this corresponds to the true jet that is reported in the paper. It's on chromosome 11. And we see kind of the intermediary outputs of the algorithm. It chose a scale of 6.59, and that's quite big. Relatively speaking, that's quite big. The angle I just want to mention maybe, I centered it so that the white is 90. So we see that most of it is 90. So it's pointing in the right direction. Some other examples. So the one on the left is another jet on chromosome 11. Its scale is it's a bit smaller. We also have chromosome 6, and these ones are a bit are smaller even still. So we have a sigma that's about 2, 3, and maybe this one's a little bit more thicker than the 2. And so we see the benefits of the multiscale approach kind of working here. And these are all top 10 out of around 80,000 ridges that were given by the ImageJ. That being said, we still have to do a very kind of comprehensive genome-wide evaluation, and that is in progress. So that's the multi-scale part. We did encounter some challenges in Hi-C data. So Repli-HiC didn't really have these TAD signals that were quite as strong as the Hi-C. And so if you see what's happening, the scale that was identified was 9, and so that's very big. So it's essentially saying that this entire pad is a jet when it's not. And so this is some challenge that we have encountered. So TADs are being recognized as jets. And so the idea is capture these TADs in scale space and then impose further constraints on the saliency. So this is currently in progress, but what we plan to do is we have the positions, we have the gamma, and so along that position, I can then compute the scale. So it begins quite large, and as we go up and up, we go through the TAD crossing point, which is kind of like a bottleneck. Here, we expect the ridge size to be quite small, and we see that kind of reflected in the scale space, and then we see it increasing back again. And so because TADs occur at a specific angle, 90 degrees, we should be able to find these in scale space. We're still doing this, this is still in progress. And then because we know that information, then we can then impose that in further constraints on the saliency measure by saying that not only do we have the XY to be oriented as a jet, but shouldn't resemble a TAD or stripe. So these are the references. And with that, I would like to end my presentation. Thank you very much.
Across the diagonal, do you expect it to be kind of like an even function? Do you ever have like odd function? Like for instance, you have them coming out like this, but could they kind of loop back around and go across each other?
I talk about in vivo, like actually.
Well, I guess in the data that you're seeing.
Typically, I don't think I've seen that kind of happen. It seems to be relatively straightforward, as in it's just like a secondary diagonal, roughly about 45, but there's a bit of curvature, but going back towards the main diagonal, I don't see.
So I guess in your along your ridges,
yes,
you don't have any sort of assumption or bias towards having them come off the diagonal at on the same side, or they're going to curve in the same direction.
In the same direction.
What in your X across the diagonal?
Yes.
It looked like sometimes they were perfectly diagonal and sometimes they curved.
Oh, yes.
But I can't picture how you would get it to curve one way and then curve the other way.
The ridge detection algorithm.
What's that?
Are you asking
in vivo? And if it does
in vivo.
happen that it is only going in one direction, your algorithm isn't taking that inductive bias into account.
So the ridge detection algorithm doesn't impose any kind of assumptions about the direction. Yes. So it will just simply link anything that has a high second derivative at that point locally. So really, we have the opposite challenge where it's sometimes linking noise together. And so we have to get rid of those cases. But in vivo, I have not really seen cases where they begin at a very kind of strong angle, if that is the question that you're asking.
And these matrices are symmetric, so.
Maybe I can ask on that.
Okay. So yeah, sorry, yeah.
Do you have any experimental evidence that the jet is really existent? I'm not sure. I mean, there are a lot of structural variants in the Hi-C data.
Yes.
So how can we say, yeah, it is jet, it is the cat, it is
Great question.
DNA? How can we say that? Experimentally or theoretically?
So experimental work has, I think I can just. So a lot of depletion studies have been conducted. So you essentially deplete, and this is especially the cohesin model, you essentially deplete certain proteins with the cohesin model. WAPL, which is the thing that unloads the rings. You also want to delete, not delete, but knock out CTCF and combinations the two and see what happens. So it does support the existence, but you're right, these are all bulk, they're not single celled. So there is always that question, but they have also been confirmed in the replication studies, which we know mechanistically that it will produce these structures. We see that in the Hi-C contact map. And there have been some polymer simulation models that show this too. Thank you.
Can I go back to page 70, like the last.
Oh, 70. Yeah. Yes.
So I just wonder, like, what's the meaning of the blue line? That's how this is detected.
Yes, that's the thing. I wasn't very clear on that. Yeah, that is the detected line.
But why there are like such thin lines? Because look at the structure, there should be like wider.
Yeah, exactly. So so the output I show plot here is just the X and Y position. So it is it has no width at all. But we can infer the size by the scale at which it was a maximum, which in this case, and well, the median is 5, 6. So that's the width roughly of the jet. Yeah.
So it means like the width of the jet.
Yes.
You need to decide that.
Sorry.
You need to define the width of the jet.
Yes, I mean we have the algorithm does give us this like an estimate of the width. So we don't. It's just a matter of plotting from now. I just haven't plotted the width onto this, but the width is about 6. So I go around 6 pixels in this direction and 6 pixels in the other direction, and that kind of gives me the overall width of the ridge.
Actually, are they wider?
It's roughly that, yes. There's an equation actually that kind of links the actual width and the standard deviation of the Gaussian. So it seems maybe it's not right, but yeah, I mean, it's estimate.
Yeah, another question. So, in the background, you talk about the speed is kind of a factor, right? So, what is the speed of this entire, like the composed algorithm?
So
The speed.
I missed the first part.
Speed. I mean, so in the background, you talk about the speed, which means the time to take.
Yes. Okay. So cohesion looping is actually very quick. It's on the order of minutes.
Minutes. Okay.
Yeah, it's on the order of minutes. So people have done live cell imaging to visualize kind of very small, so not genome-wide, but really very small local parts of the DNA, specifically the cohesion looping events.
So it's much faster than previous methods, right?
Oh, are you talking about the complexity, the speed of the algorithm?
Yes.
Oh, sorry. Yes,
No.
I misunderstood. Yes. So we don't do any polymer simulation to get the kernel. So this is just convolution.
Okay.
That being said, we do a lot of convolution. So we have to test this also, but the preliminary tests were all under 30 minutes for the whole pipeline.
Okay.
Another question. In your background, you had these other methods that were able to classify regions as being jet, not a jet.
Yes.
And yours also is a classifier, saying
Yes.
this is a jet or not a jet.
Yes.
So you have these data where people hand annotated some examples of them.
Right.
You can use that as sort of a gold standard set. But I'm curious, in evaluating these classifiers, how should I think about the cost of missing some versus calling some that are not true? The sort of false negative, false positive trade-off.
I see. So I think that would really depend on the data. So if we have something like Repli-HiC, which has relatively straight jets, as is in this example, then something like the previous algorithms that assume that jets were straight, that jets were at 45 degrees, those methods I think would do quite well. So when it comes to actually kind of finding that position boundary, I think we could be fairly confident that these are, because we have multiple methods to test them, we can test them all on the same data set and then see the overlap. But for cases where the jet sizes and shapes are quite variable, in cases where you have asymmetry, I think that will then mean that this algorithm should be able to find those jets, and so we would see a difference in the true positive, false negative. As of actually finding the threshold, that is something that I would have to look into. Yes, like a threshold to actually kind of say, is this a jet or not?
Maybe I can ask it a slightly different way. If you, let's say you had an experimental cloud writer and they said, please find me all of the jets, and you ran your algorithm and you gave them back a list, and they said, okay, now we're going to do an experiment based on your data. I'm going to take that, I'm going to do some complicated something, and it's going to cost some amount of time and energy and dollars to test that. I don't have an intuition about what they would even do with these once they found them, and why it would matter to them, and why you wouldn't want to say find them all and have a lot of false positives, or you want to find just a very few that are really, really high confident. Like I guess I don't understand what you would do once you found these structures.
I see. So the ultimate goal, maybe.
Yeah.
Yes, yes. So I think really the functional implication of jets is not really understood. So I'm not so sure about that exact scenario where it's going to cost a lot of money, and we don't know really what to do with them. What is the use case, and that would kind of influence my decision on the true positive, false negative. Yes, so this is, I think, at this point just almost exploratory. As of the cohesin model, we're not really sure functionally what exact impact they have. They have been associated with originating from small active A compartments, but also in quiescent cells, so cells that are not undergoing the cell cycle. So it's a bit of a mystery as to why they happened themselves and so late in the Hi-C. Why weren't they in 2014 when I'll publish the in situ Hi-C with a very high, deeply sampled maps? Why weren't they there? So I think we're just not really understanding the functional implication, and an algorithm such as this, that can characterize the different shapes, that can track jets that go through different angles, would be of help in a more like exploratory way, but not so much as to say we have extreme confidence in that these are the jets for what. Yes, we're not so sure. Yes.
You said that this didn't know information why
So why?
Yeah, so,
Yes, good question.
yeah, why do we see all these different
Variations? Yes. So at least within the cohesin model, the variations arise from it would be like what I would guess to be asymmetric CTCF. So if I have strong CTCF binding on one side, but not on the other, then that would cause the one side to be kind of slightly tilted towards. And sorry, I should mention this. So some of these studies didn't see jets in wild type. They had to do a knockout. So I'm assuming that you then knock them out. So you do the CTCF knockout. And in an asymmetrical case where you have CTCF different, whether the two CTCF that you have is asymmetric binding strength, and that is one example I can think of where you would have an asymmetric condition and you do the knockout. But again, I think for the other cases, why do we see asymmetric conditions? I'm not so sure. I think that is tied to do with the understanding of functional implication. Thank you.